some of you may remember we actually had, I think the first time we've done this was last year. It looked a little different. One, because I think it was like 8 a.m. What is the equivalent of tomorrow, which I think is tough to make. And also, we didn't have slides or anything, but those of you who were there, this talk may be a little bit similar, but I hope you can still find some use for it. So I should introduce myself. My name's Eric. I'm from the lab of Manash Patel. Many of you will, if you don't know him already, you'll see him tonight at the gathering. And he has a lab at the University of Virginia. I'm a graduate student there in my partway through my fourth year. And so I'm really thankful that the Syndrome Foundation, SCN8A field, was able to invite me again. I'm just really honored. My hope for this time, for this talk, whatever it turns into, is just to be helpful to the families. The task that was given to me is to try to explain a little bit of the basics about the SCN8A encephalopathy, a little bit about neuroscience, how the sodium channels work. So if you've ever had an introductory biology course, this might feel a little bit similar at times, but my hope is that by the end of the session, you feel like you've gained something about how, of what's going on in the context of sodium channels, neurons, how do brains function. And so this talk, I have a structure, we can go through that, but at any point if you have a question, please just either yell it out or give me a signal or something, because I want to go down the tangents that are the questions that really are coming from you. In our lab, we recognize we study SCN8A in the lab setting every day, but we'd be the first to admit that the real experts on SCN8A are the families, and so I acknowledge that and just am thankful for the time to share what I can. So, without further ado, SCN8A 101, and so what I will try to cover today are a little bit about, we'll start at the brain, talk about just really basic, what does a brain do? What is a brain designed to do? And talk about it being a super complex master organ of the nervous system. Then we're going to zoom in and talk about neurons, which are the specialized excitable cells of the nervous system and of the brain. Zooming in even further, we're going to talk about action potentials, which you can think of as the language of the nervous system. And then zooming in even further, we're going to talk finally about voltage-gated sodium channels. So that's right at the heart of SCN8A. We're going to talk about how they drive action potentials and how that relates to the context of epilepsy. And then I will hopefully have some time to just take questions that you have. That if you've ever had a question you wanted to ask someone in the research community, I would hope that this is the safest place for you to feel comfortable to do that. And I'll do my best to answer. Two other notes. One is I'm not a clinician. I'm not trained in the medical field, and so I will do my best to answer, but there will admittedly be times where I cannot give you medical advice, and so that's really important. And then the other one is sometimes people include a disclosure slide. I didn't put it in, but our lab has worked with Praxis Precision Medicines in the past, just to be transparent about that. So, okay. So when we think about a brain and what brains do, epilepsy is a property of the brain, so it's kind of important to start there. And I'd like to stress to you that we can think about the brain as a very complex input-output machine. So it's in a lot of ways similar to a calculator, to a computer, in which you have some sort of input, whether that's typing or clicking on something. The machine does some processing, and then it executes an output. The brain is designed to take in information from your senses and perception, so that would be like your sense of smell, your sense of touch, your sense of body awareness. It performs a computation or processing, and then it brings also execute some output. So speaking or movement are the actions of the brain. And so I say this just because sometimes thinking about brain function is admittedly very complex, but to make it as simple, to think about it simply, it would be to think of it as an input-out machine. And so I already said that it's highly complex. Your brain is comprised of around 100 billion neurons, and as well as many billion other cells. And so to study the brain and understand the input-output relationship that the brain functions with, it is highly complex. So I guess I'm giving both sides of the spectrum. Let's think about it simply. Let's use the idea of an input-output to help us get started, but admittedly, this is very difficult to understand. It's not only difficult to understand because it's complex inherently, it also changes itself. The brain is, you may have heard the term plastic. It is able to adapt, which is the foundation for learning and memory. You actually, you're not the same person you were last year, and that's because the brain is constantly adapting to its environment. And so, as researchers, when we try to understand brain function, or when the brain dysfunctions, we're trying to understand in that chain of input, processing, and output, what is going awry and how we can restore it. So let's zoom in a little further and talk about neurons. So neurons are specialized cells of the nervous system. So nerve cells, neurons, they're the same thing. And they're specialized in that I'm going to get to this, they are excitable, which means they're electrically active. And we're going to spend some time talking about action potentials. But I should highlight that in the same way that the brain has a direction of information flow in that senses, processing, output, neurons themselves have that same directional flow of information. So neurons have three main structural components. This is a cartoon of a neuron that I had a student of mine put together. Here at the top, you have this part called the dendrites. They receive the signals from either other cells or from some sensation directly. So like all along your body, you have touch sensors. Those are the equivalent of taking in some signal in the dendrite. That signal then goes to the cell body, which integrates that information. That's just another way of saying gathers up all of those signals, and then in the axon of a neuron, it is able to send that signal forward. And that signal, I know it's a little abstract, that signal is encoded in changes in voltage. Now, if words like voltage and excitability start to kind of like freak you out a little bit, hang with me, and I'm hoping we can gain a little bit more of an understanding of what that means. But for now, just know that neurons are specialized in that they are electrically active. They are excitable. Those terms have the same meaning. And so this is what makes neurons different from skin cells, different from bone cells. Most cells in the body don't have electrical properties that use electricity as a signal to process information, but neurons do. When I say excitable, that's another way of saying it is that they use the flow of positive and negative charges. Charge carriers are ions, and so that's where we're going to talk about sodium and potassium. So any questions there? I know we took a couple steps. We talked about brain simply, and now neurons. Okay. I want to pause for 1 second and think about why does a brain use electrical signaling at all? And the real point of why nervous systems evolve is to do things quickly. Voltage changes can happen very, very quickly compared compared to, for example, you have signaling in your body that's not electrical, maybe hormonal or something, but they function on the order of minutes, hours, and days, whereas in the nervous system, you're able to respond to an input and orchestrate an output in a matter of milliseconds. Just imagine a time when you picked up a cup of coffee that's too hot. You will let go within a matter of milliseconds without even processing it. And this is a critical function for organisms to be able to survive. And so electrical signaling in the nervous system is very critical to survival and is very fast. And if you have a fast signaling, you have to have whatever the signal itself is, has to be very, very fast and very precise. And so nerve cells, neurons, use action potentials to accomplish this. And this is a quick spike in membrane voltage, or you can just think about it as the electrical potential energy of a cell. Quick spike that looks something like this. I know this is a little bit dim. You have and I'm going to spend more time on this, but just this blip in membrane voltage is the signaling event that neurons use to communicate. And I also included here that not all action potentials are the same or identical. The diversity in neurons is recapitulated in diversity in the action potential. And why this matters is because if an action potential changes even slightly, the information that it is carrying also changes. So you can, maybe a metaphor would be, we have many words in the English language, but not all of the words carry the same meaning. In the same way, action potentials, which are the language of neurons, capture the meaning in the shape and the form, and how many of the action potentials they set. And again, we're going to spend time on this, but I wanted to stress that this, even though I'm speaking about it simply, this is still a very complex thing to study because action potentials vary widely throughout the brain. But all neurons have in common the ability to initiate action potentials and signal through action potentials. So again, this might be a good time. Any questions? I hope you're still with me. Action potentials, fast electrical signals in the body. But neurons don't function just by themselves. Since you have 80 to 100 billion neurons, they function together in networks. And I have you can see, this is just a graphic depiction of multiple neurons sending an action potential throughout multiple, in the network, kind of like an electrical signal passing through a cable. So they function in networks. So why does this matter? Well, it matters because seizures and the disorder of epilepsy is fundamentally a network disorder. It is a dysfunction of neurons together. So you can't have a seizure at just the single level of a neuron, but you can have a seizure at the level of groups of neurons behaving in networks. In particular, seizures are when neurons individually become hyperexcitable, and then in the network they begin to synchronize, in that they fire action potentials at the same time repetitively. This matters for those of us who want to try to understand what epilepsy and seizures are. Is that for me? That is really great. Thank you. Okay, great. So now I don't have to project quite as strong. So the hypersynchrony is the hallmark of seizures, and so I wanted to say this because the task of maintaining proper balance in the brain is much like tightrope walking.
because the brain has to keep excitability going at all times. That's what we use to think, integrate information from our environment and live. But it also has to keep itself from entering into a seizure-like state. And so this balance is very precise. And with that, I've tried, I don't know if everyone in epilepsy research would agree, but I think one way to think about epilepsy research is to boil everything down to 3 questions or the major aim. Why do neurons become hyperexcitable? How do networks become hypersynchronous? And what can we do, or how can we restore the balance? And so yeah, if you're taking notes, that might be one. I don't know if one to write down, but I think that helps give us kind of a framework of thinking about how to study SCN8A encephalopathy, because we're able to study it at different levels to try to get at these questions. And so I'd actually encourage you as you watch and listen to the science talks tonight, which admittedly may be challenging to follow, see if you can place each of the talks in one of these categories. Though I would propose that they could probably each be put into one of these categories. They're focusing on neurons and why they become hyperexcitable. They're focusing on seizures and what's going on in the network. Or they're trying to understand how to treat the disease. Okay, questions now. Okay. So let's talk a little bit more about action potentials. We're slowly making our way to voltage-gated sodium channels. I think the best way to do that is to take a look at the action potential and what do action potentials have to have in order to be an action potential. And I think it's good to know this because action potentials are, I'm calling it the language of neurons or the grammar of neurons. They're what contain information. And so fundamentally, the action potential can be broken down into 3 parts. First is the resting phase. And I'm plotting it with this example, and this is membrane voltage, or just the electrical energy of the cell. At the resting phase, the neuron is not doing anything. It's stable, and it's able to respond if there is a signal. So this might be like a touch receptor right now that you're not currently pressing on. It's poised, it's ready to respond to information, but it's not currently carrying any information. That's the resting phase. The second part of the action potential is the rising phase. The membrane potential rapidly rises. I haven't labeled the time scale here, but this is on the order of milliseconds. So thousands of a second, this whole thing will happen. So the rising phase, the membrane potential really quickly goes from this lower value and skyrockets up to a much higher value. And then the falling phase, it drops off and returns eventually to resting. And so these 3 parts have to happen in this sequence to initiate an action potential. Rest, the rising phase, and the falling phase. And they have to happen very precisely and quickly to properly encode the information that this action potential is sending downstream to either processing or to some output. So it's kind of simple. I hope this is as simple as you've seen it. So it's rest, rise, and fall. 3 phases. But just like any device that we use that uses electricity, neurons actually have to have some sort of power source, either a battery or some way of setting the system to respond and use electrical energy. And so the one that does that is this pump in the membrane. So I should back up and maybe I can play that again. This red box is zoomed in on this neuron's membrane, right on the edge of the cell. And this protein is called the sodium-potassium pump. And it just sits right in the membrane. And it helps get the cell ready to fire an action potential and signal. It is the battery charger for every single neuron. Just to put this in perspective, the estimates are about 1/3 of all of your food energy that you eat, that sustains your body. 1/3 of the energy from that food runs this pump alone. This is a really important protein for survival, sodium-potassium pump. So I'm going to play it again. So when it runs, it's going to push all the sodium to the outside of the cell, and it's actually going to pull in the potassium to the inside of the cell, just like that. So why does this matter? Why would it matter to have sodium on the outside and potassium on the inside? Well, let's think of it like a dam. We build up a gradient for sodium and potassium in the same way that the Hoover Dam has a gradient of higher water on one side, lower on the other side. Now, since we've established a gradient, all you have to do is open a gate, and in our case, sodium will flow into the cell, or in the case of the dam, water will flow through the dam. And this matters because we've charged the system, we have our battery creating that gradient, we can determine how much water gets through the dam by controlling how we open the gate. You can open up the gate to be very large, you can open it very quickly, slowly. You have a lot of power in determining the amount of water that gets through based on the gate. And in the exact same way, we've established a system where we can allow sodium to go into the cell very precisely. Okay. Any questions? Is everyone with me? Question?
How does it travel back? Once you get all the outside?
We're gonna get there in the next slide. So this is the battery charger, and then I hope with the next slide, some of these pieces that I've established will come together. I'm hoping. If it doesn't come together, stop me, because this is right at I think it should be probably the most challenging part of the talk, but if you're with me, I think it'll start to come together. So we've set the stage for the action potential. We needed to do this so that we can precisely allow sodium in a way that we can do work with it or signal with it via the action potential. So voltage-gated sodium channels are proteins that also sit on the membrane of a cell. So I've zoomed in again here. So you have the sodium-potassium pump, but you also have voltage-gated sodium channels. And they're gated in the same way as our Hoover Dam metaphor. They're gated with two separate gates. You have one activation gate and one inactivation gate. You don't need to know what distinguishes them yet. Just know that if a single gate is closed on the voltage-gated sodium channel, the sodium cannot flow back into the cell. So for example, channels are closed most of the time. And if it tries to flow in, it doesn't. I'll show that again. So sodium can't get into the cell. The sodium channel is closed. However, if you open up that activation gate in a process called logically activation, you can get to an open channel. Both of the gates are open, and now sodium can flow into the cell. And it does that for such a short time because it turns out that this, when both the gates are open, it's very unstable, and very quickly after it, in a process called inactivation, the other gate closes and stops the sodium from flowing. And so, so no longer, this is inactive. So we have three unique states for voltage-gated sodium channels: closed, open, and inactive. Now I hope that this is kind of starting to sound parallel You have 3 phases of the action potential that have to go in sequence. And now you have 3 phases of the voltage-gated sodium channels structure that allows sodium into the cell in a very precise way. So it turns out that the upstroke of the action potential is caused by sodium flowing into the cell. So these are 2 ways of looking at the same process. So now let me just connect it for you. So voltage-gated sodium channels, I'm going to show the same depiction of what's going on with the channel and now connect it to what we were talking about earlier with the action potential. The signaling event for nerve cells requires that sodium channels first start out closed. So I have an arrow here, it's not showing up very well, but right now we're at rest, our membrane potential is stable in the whole neuron, and most of our voltage-gated sodium channels are closed. When some stimulus comes by, it opens the channel in a process called activation, and that sodium flows into the cell, causing the membrane potential to rise. And then really quickly, again, all this is happening in millisecond time scale, the channel inactivates, it shuts that other gate, and the membrane potential falls. This is the falling phase, the downstroke phase of the action potential. And what I want to stress now, so this cycle of sodium channels is required for the ability for a neuron to cycle and signal properly. If a sodium channel gets stuck closed, you cannot initiate an action potential. If it gets stuck open, you can't recover from an action potential. And so, for the research community and the way that we understand voltage-gated sodium channels is we study in very sophisticated ways basically the simple progression from sodium channels from closed, open, inactive, and back to closed so that they can initiate this process over and over again. And it turns out that when voltage-gated sodium channels, if you change the properties through which they go through this process, you can lead to very devastating consequences for the action potential and ultimately for neurons functioning in networks. So this is a foundational element of the SCN8A story, is that voltage-gated sodium channels are very, very precise and require going through this process in a way that is highly regular. It has to be precise. This makes sense because you have 100 billion neurons, and you probably, I don't even know the number, but you probably have millions of sodium channels on each neuron. And so this is a process that even a small tweak in this process could have very large consequences. So we started with brains, went to neurons, we talked about action potentials, and then voltage-gated sodium channels, and now I've hopefully connected the two. That the signaling event of neurons is driven by voltage-gated sodium channels. These are what a cell that has voltage-gated sodium channels, they're excitable cells. If you don't have voltage-gated sodium channels, you're not a nerve cell. This is part of why SCN8A and NAV 1.6 or other voltage-gated sodium channels are so critical for function of the nervous system, because they're part of what makes the nervous system the nervous system. Okay, any questions at this point? Yes?
Is the pump linked to this, or does the pump always pump out?
That's a good question. So the question is the pump linked to this, or does the pump always go? The pump is always going. In fact, if you block the pump, it's a toxin. If you stop the pump, you would actually get seizures, and if you block it too long, it would be lethal. So it's the pump is absolutely critical, and it's always going. Any other questions? Sure. That's a good question. I conveniently didn't talk much about potassium. So potassium will also leave the cell. There are more channels. They're actually what's called leak channels. So potassium is actually less regulated than sodium. They can pass in and out. Sodium is highly regulated. You have pretty much all the sodium that flows through the cell is going through either the pump or the voltage-gated sodium channel. That's a good question. Any other questions while we're at it? Yeah.
The inactivation gate plug, it looks in your graphic as though it's not completely closed. Is it just the graphic?
That's very. I actually didn't even design that, but that's.
And does that correspond with the slower slope back down?
Yes, that's a great point. So the slope here is slower than the rising phase. This is, and that's in general. Yeah, I would actually agree. Yes, I have never recorded a neuron where the downstroke is faster than the upstroke. There's a number of reasons for that. For brevity's sake, this is a substantial focus of ongoing research in SCN8A, is that it seems like many mutations that we've characterized hit this process, and they make it, the channel doesn't quite close all the way. It turns out in normal cells, it's not perfect. There's about 1% that can stay open, 1% of the channels. But if you dial it up even to 3 or 4%, it can be very severe. I'm going to show actually a few examples in a moment here, how that would affect the action potential. But that's a great observation.
So sodium and potassium both have a charge of plus 1. So obviously not the same amount of sodium and potassium are being changed through the
They have,
action potential.
yes.
Difference in voltage off the brain.
Yes. I'd be happy to go into details of that probably after. Yeah, it's slightly more complicated. It turns out the pump pumps 3 sodium out for every 2 potassium that go in. So there's a way that the neuron establishes and maintains a non-zero potential. Okay, so what about SCN8A? I almost got through the whole talk without talking about what SCN8A is. So, and I know many of you know this, but just to be clear, so SCN8A is the gene that codes for the NAV 1.6 channel. So this is the NAV 1.6 channel. The SCN8A is a gene. We can think about the relationship between SCN8A and 1.6 as being the instructional manual to build the 1.6 channel. So the SCN8A gene is instructing machinery in the cell to know how to make this protein. And so mutations in the SCN8A gene is just another way of saying the genetic sequence or something about the gene has changed such that the instructions to build it, when read as it's sent to the machinery, changes the shape or the structure of this protein. And why that might matter is because the channel needs a very precise shape in order to go through this process with high fidelity and precision. So mutations in the gene lead to a different protein, a different channel that may function differently. Part of what we do in my lab is a very simple experiment. We just take mutant copies of SCN8A with different mutations and try to understand how they go through that cycling process. What is different about them? Whatever is changed in a mutant 1.6 is likely to be very important for epilepsy and for understanding how the excitability of neurons is changed in the condition of SCN8A encephalopathy. So just again to highlight, SCN8A is the gene that contains the information for building the 1.6 channel, which is required to have nervous system, voltage-gated sodium channel cycling, and excitability, firing of action potentials. Questions here? Okay, we're still going to have plenty of time for questions, but so what's unique about SCN8A and NAV 1.6? So this gets at that first question in my three categories of how to understand epilepsy. So how are neurons becoming hyperexcitable? How are they becoming too excitable and leading to seizures? Well, 1.6, the SCN8A gene, turns out that many of these channels, these proteins, these voltage-gated sodium channels, sit in the membrane at regions that are really important for where the decision to fire an action potential is made. So they determine for the cell when to fire an action potential, or how many action potentials to fire, as well as the shape of individual action potentials. And that matters, like what I showed you earlier in the presentation, because neurons that are different change their shape of the action potential to show their uniqueness. And if SCN8A changes the shape of the action potential, it may affect the message being sent by the neuron. It may affect that signal somewhere in the process. So this is text. Let me show you some recordings that I've done that show this. So I'm an electrophysiologist. That's just a long word that says I look at how individual neurons function and how they fire action potential. So these are actual recordings that I've done, and you don't need to follow the specifics of it, but I've highlighted here that if I ramp and I cause some stimulus for these neurons, normally wild type NAV 1.6, you have to get further and then they spike. They have action potentials. But when I do the same thing in a mouse model that has a mutation in the SCN8A gene, which leads to a dysfunctional 1.6, that AP, the action potential, initiates too early. Initiates well before wild type. So you can think of this as being, it's a little bit extra sensitive. It begins to send a message even before the message is ready to be sent. Here's another example. If I do a similar stimulus of this kind of rectangle pulse, I can get a whole bunch of action potentials in a row. And if I count them, I find that, I should have listed the number here, but in general, when we record from neurons that have mutations in SCN8A, they fire more action potentials. No matter what stimulus you give them, they fire more. You can think of this, instead of being hypersensitive, this is like turning up the volume on your stereo. Every message that is sent might be sent too loud. Or it could even change the meaning of the message being sent, of the signaling. So NAV 1.6 is important for how many action potentials. And one of the most striking things we found in the last, in the course of our last few studies, is that NAV 1.6 is very important for the shape of the action potential. This is a recording in a wild type neuron, and this is a recording from a mouse neuron that has a mutant copy of 1.6. And I hope it's obvious to see, even for someone who's not a scientist, these are very different in terms of the shape of these events. They have this kind of burst-like firing pattern, where these are kind of, from one to the next, are kind of pretty much the same. It comes, cycles, comes back, it fires, comes back. These, on the downstroke phase, before going back to rest, it actually reinitiates a kind of spikelet. And this gets right at the point, the question earlier that you asked her was, why, what about the downstroke? How is that affected? This is exactly, we think, because channels are not inactivating properly, they actually can flip themselves back open in the mutant condition, and this leads to a very different signal in a brain region that's really important for epilepsy. It turns out that if you put a sodium channel blocker on this strangely spiking neuron, it can actually block just that second spiking. This together is evidence to us that this might be really important for how neurons become too excitable. And this might be something that we can think about when we're trying to understand how the mutations lead to changes in excitability. So again, just to highlight that, NAV 1.6 does many things. These are not the only 3 things that this channel does. It's very wide-ranging. We're trying to understand that. But we have already made some progress, and what we found is that NAV 1.6 is important for when to fire an action potential, how many, and the shape of the action potential, which is important for the message. And so overall, SCN8A mutations can result in a process for the voltage-gated sodium channels. Their cycling is affected, which affects the action potential firing, which affects, multiplied over 100 billion neurons, how those neurons function together in networks, and can ultimately result in epilepsy. So just to summarize, this is the end of the talk, and then we'll have plenty of times to just ask questions if you have them. The brain is incredibly complex. It can be thought of as an input-output system, but it's difficult to fully understand exactly what's going on. It's specialized to quickly take in information, respond, and that is accomplished through neurons, which are the individual special excitable cells of the nervous system, and they communicate through these changes in membrane voltage, action potentials. Action potentials are driven by voltage-gated sodium channels, like NAV 1.6. They're specialized to accomplish this task, and even a small tweak in the properties of a voltage-gated sodium channel could result in some behavior that is not normal, that could be really important for epilepsy and the mechanism for SCN8A encephalopathy. So with that, I always need to acknowledge that I work with a team. They're excellent. Our leader is Dr. Manoj Patel. We have other members of the lab, including some of my undergraduate students who work with me, namely Sammy. She's the one who drew some of the cartoons for this presentation. And with that, I have my email here. Don't hesitate to reach out. One of I think the most magical things about the SCN8A gathering is that for scientists like myself and my colleagues, we rarely get to hear from parents who are the experts of what's going on in the lived experience of SCN8A encephalopathy. It's not only inspiring to us to hear your stories, it's also part of how we generate ideas for moving forward. So through tonight, or if you ever have something you notice and think, man, that's kind of interesting, don't hesitate to send that to us. We're thankful for that. And with that, I'm just, again, very grateful to have the opportunity to speak with you and ask any questions you have, and I hope I can answer.
Thank you, by the way. So the sodium 1.6,
Yeah.
a lot of our kids have cardiac stuff too, or the fear of cardiac stuff. So nothing on the cardiac cells, or is it like the neuronal input into the cardiac cells that we worry about?
Yeah, that's a great question. So the question's about what's the cardiac role of 1.6? Admittedly, I can't speak to that very well. I spend my time focusing on cells of the nervous system. Cardiac cells are excitable. They express predominantly a different sodium channel, but there has been, I know groups have suggested that 1.6 is there. But you can imagine that it could either affect it directly, it could be in the heart, or it could be from the central regulation I do know that groups are looking at that, so we're trying to understand.
Sure. With the mic.
Have you ever come across a case where you may also have anti-thrombite deficiency?
I don't know exactly.
Antithrombin 3 deficiency. I'm not familiar with that. But I'd encourage you, if other folks know what that is, or maybe a clinician. I think I'll just pass around the mic. I feel like that would be easier.
So I know you said that neurons are part of the nervous system, so like even touch sensors. So our daughter has really high pain tolerance, so like doesn't recognize that maybe something would be hurting her. So that would be part of these cells not functioning properly.
Exactly. So the pain system is it uses the same cells of the nervous system as functioning in the brain. So you can think of this exact thing kind of as some disconnect in input, processing, and output. That is something that we've heard through the gatherings that there might be many patients who have either pain, particular sensitivities, or a lack thereof. And so that's something that I don't know if anyone's following after, but I'm sure we all have the interest in following that because that is a really important part of the story.
And would that link to like them knowing they're hot or cold?
Yeah,
Okay.
temperature sensitivity. So, yeah, they're all part of the same system.
What about all the developmental challenges that come with, even in the absence of seizures, like even when seizures are controlled, all the other developmental issues like speech and ataxia, et cetera? How do you guys think about that?
That's a really great. Did everyone hear that? The developmental aspects. That's something that is always, I think, going to be challenging for the field. Is to what degree are seizures causing the developmental challenges, and to what extent is the developmental challenge something that happens just from the channel functioning differently? Because they're probably both happening in some mixture. Severe seizures can impair development. So you mentioned ataxia. That could be something that is could be specific. Ataxia is usually associated with cerebellum dysfunction, and as far as we know, the seizures don't hit the cerebellum. So I think there's a lot of the clinical, a lot of the spectrum of SCN8A patients. I think we still have a lot to learn. Yes.
Always complain that she has the group moving like vertical or vertical natural out of the lungs. Is it also related?
Could be.
Could be, but we cannot say for sure.
I mean, I can't. I'll remind everyone, as a graduate student, I won't be able to give much insight into particular clinical, but these are exactly the kind of things that, as the research community, are very interesting to us. If someone says they have dizziness, or their auditory sensing is really sensitive, or not so sensitive, their hearing, these are all pieces that we're interested in. I can't say it could be. The vestibular system functions through the nervous system, and but it's also, dizziness is a very complicated clinical presentation to try to understand. Thanks.
Is it possible to see a show of hands for how many people see the lack of pain?
Yeah, I'd love to see it.
Because we definitely experience
Lack of pain? Okay.
Oh, yeah.
You know, they have a very high threshold.
Okay, now, hypersensitive to pain. No one.
That's interesting.
These are exactly the things that really are interesting to the researchers. So thanks for that.
Mine's a
Little sensitive.
hot, cold water.
Oh, yes. Yeah.
Okay.
But I mean, if he hurts himself,
He has broken bones and not even cried.
Yeah.
Yeah.
So when he does cry, I know it's something extreme.
Yeah.
It's interesting hearing about the different levels of pain tolerance, but also Will just got startled really easily, and I think other people have mentioned that. So that sounds it sounds actually opposite to having a high pain threshold, but I think we talked about like a strong startle reaction.
My response to
Sounds, and I know some of the kids have actually had seizures out of having a strong startle reaction, you know.
With sounds.
Sounds in particular, yes.
Something really. Can I get a show of hands?
Can I get a show of hands of sounds causing a seizure? Or causing a seizure or just a really severe startle response? Wow, okay, so that's something that's really important, I think, and really interesting to us.
So my son has, I guess he's his seizure types are myclonic, and we thought he had infantile spasms, but they were myclonic seizures, so he did have a lot of startle type, and he does he's very sensitive to light.
Very sensitive to light.
So, yes.
Anyone else sensitivity to light? And now we're kind of like almost like survey, but and this is the kind of thing. I mean, the registry might have a lot of this information. So love it. Okay, great. So this is exactly the stuff that you've worked to compile that is really helpful to us in the field. So thanks for that. Yeah.
Is there any correlation with how the different variants affect the way the NaV1.6 activates and deactivates?
I would say that trying to relate the biophysics of the channel to the specific mutation, relating the function of the nerve cell to the biophysics, and the function of the network to the neuron is the holy grail of epilepsy and of SCN8A, and that's what that's been the focus of my PhD. It's a difficult thing, but we're making some progress. The idea of to fully understand what's going on is to understand the input-output machine with perfection. To understand and then understand how to restore it. So we don't yet have the full picture. I think we're beginning to make progress understanding certain patterns with where the mutations are, but that requires a lot of ongoing work.
So I have a question, so Saki's dad has the mutation, but he just did that, but he's perfectly fine.
Yeah.
So then is that what?
Do you have a better answer to that? Why?
Well, there are about 10% of families where the mutation is inherited. And sometimes clinicians will say, well, that means it doesn't cause epilepsy or doesn't cause issue. We know that's not true because we don't have one gene in isolation. We have a whole genome, and there are lots of genes that work together in all of the things that are discussed. And so some genes might have variants that compensate for a variant in another gene. And that happens quite often. And that's really on the cutting edge of where research is at. We don't know how to combinatorics of all the genes and all the variants. Very difficult to understand. But given that this is a recurring theme, we come to believe that SCN8A is probably playing a role in most everyone's phenotype, even if the parent doesn't have it, because they may be protected. And there are variants in SCN8A that do not cause issues, and we're trying to find those regions of the gene where we might find those. And so that's a very active area of research. It's very important.
And one that you're always can answer, which is great. Our lab is less to do that.
More questions?
Does the potassium come in and out the same as the sodium? It's through the same
It's not through the same channel. They have their own. Potassium has their own channel, so they got a separate regulated system.
So it's coming in normal, and then the sodium's coming in not correct.
Yeah. The challenge is they're related because the sodium, because of its ionic charge, actually affects the potassium function. So So changes in a voltage-gated sodium channel are very wide-ranging. They affect things beyond just themselves. Everything in the brain, everything in their cell function is related.
So is there a seizure med that, maybe potassium type seizure med that may help?
This is a great question. The question is, could you treat SCN8A with a potassium channel modulator? I think that is something that we need to explore. I think it's ongoing as well, so it's a different strategy than what we've had, and I think that's what we need.
Yeah, because some of them, the other ones aren't working.
Exactly.
Yeah.
We have 3 children that all have the SCN8A genetic mutation, but only one of our children has epilepsy that we know of, but all 3 show different traits of the mutation.
With the channel blocker,
Would the channel blocker help
Even if-
our other 2 with some of their
If you do not have
symptoms versus just not?
versus her who does have epilepsy?
I appreciate the question. I'm going to try to artfully dodge and say that's a great question for a clinician. Where it gets back to that first point back there, that same mutation, different person can lead to something completely different. And so we really we're not well aware of what's going on. But it's a good question.
Also, we will have time tomorrow over lunch. We'll be having a working lunch with some of our industry partners who may be able to speak a bit more easily to questions like that regarding, especially specifically the sodium channel blockers that are being explored and are in development at the moment.
Okay, thank you.
Thanks. Yeah, I hate to, for any question I dodge, I hate to do that. I just can't.
We're a strange family anyway.
I do appreciate the question, so thank you. More questions? Anything? Yeah, okay. Okay. Oh, you've lost your voice. Then you need that even more. I thought maybe you were preparing to project.
I know children are all different, but why is the onset of seizures like 5 or 6 months typically? Why are they all born having seizures? If you can understand me, you're talking.
Yeah. So the one possibility, we don't, I can't say why they all start at this time. Also, since they all don't start at the same time, I'm sure there's some variability in the room, but we do know that there is some developmental regulation of 1/6. So there's a time when it begins to increase its levels. So one would think that the seizures might be related to that increase in the level. Development is a really critical part of this story, and we're, the same answer I'm giving for everything is we're looking into it, I think. And that's not just a political, I'm telling you that we are looking into what's the difference with mouse models. What is the difference between a mouse that's 3 weeks old, has the mutation but doesn't start seizing yet, and 8 weeks when they start to seize? What is the fundamental difference there? If we understood that, then maybe we could use that system to prevent seizures.
Which, if you could have any answer, right, and kind of complete your research, what would you, where would you want to go with this? What's your next step?
My next step? Well, I can tell you, I have some work that's getting really close to being published, so I don't want to give too much away. But the short story is we've assumed that, let me back up. I presented the story that there are different neurons. Well, we know that the neurons are very diverse, but one of the 2 main categories is you have excitatory neurons, which drive some function, and you have inhibitory neurons, which function as the brakes of the brain. And what we've been focusing on in the Patel lab, and what I've done, is we've assumed for much of the SCN8A research that they're making excitatory neurons more excitable. That's kind of what I told you. They lead to hyperexcitability. But what I'm hopefully gonna be able to release pretty soon is that inhibitory neurons are affected as well. And so you can think of this as I'll use a car metaphor. If SCN8A is a car that's going too fast, there's additional throttle, that's definitely true, but I'm going to argue that the brakes are also messed up. The brakes are also not functioning properly. And this matters because you may be able to treat a broken brake system in a different way than you treat a car that's just going too fast because of extra throttle. That's what I'm trying to do now, and you know, I'd love to. I'd hope that would spur on more research in this direction, because if you follow some of the literature, what I'm simply showing is that, and again, I gotta be careful not to say too much, but SCN8A and Dravet have seemed to be very contrasting, and there may actually be more similarities than previously appreciated. And I think that may give us some novel ideas to continue on our research. So be on the lookout for my paper coming out hopefully soon in 2020. So cool. Any other?
So could you speak a little more into the different variabilities that come within the voltage-gated sodium channels?
Mm-hmm.
To my understanding, it has to come down to different amino acids, amino acids being switched around when they're not supposed to. Is that a structural issue, or is that kind of like an electrochemical issue?
That's a great question. So there's more than one voltage-gated sodium channel. You have, I think the genes go up to SCN8A, but it skips a few, so I think it's around 8. The point is there are many different isoforms, and they're different in their structure. The genes are different, which has different directions to make slightly different structures, but they all function similarly, and they're similar in structure. So this might, again, I use a car metaphor. I don't know why I do that, but a Corolla and a Civic are not terribly different from a Mazda 3, but because they all function fundamentally the same, but they're slightly different. What a mutation does is changes the structure in a way that affects the functioning of that particular channel. So to answer briefly to your yes or no question of whether it's electrochemical or structural, it's structural. But because the protein's function leads to an electrochemical process, it has effects that are electrochemical. Does that make sense?
So I was hoping, I've had the opportunity to visit a couple of labs and see the rigs that you guys use to measure these action potentials, which are super impressive. And I was just thinking it might be nice for you to kind of draw a picture for everyone of what the inside of your lab looks like and the many, many hours you spend in front of these rigs.
So she's talking about what it looks like to do an experiment that I do. I'll walk through that. I'll give a quick version, but if anyone ever wants to hear more, I could be happy to go into that. But so we in the Patel lab are an electrophysiology lab, which means we spend our time using very high-tech equipment to look at the flow of ions into individual cells. And so I actually record from neurons that are in brain slices prepared from mice. And I keep the slice, this always, my parents are not scientists, and this always freaks them out. I can keep the slice, like once I take it and I slice it, I can keep the neurons alive for about 6 hours. So I have a period of time in which I can try to understand how a neuron functions. And so that's what all of those traces that I showed you are from living neurons from mice. And we have, thanks to Michael Hammer, we have a mutation that with one single amino acid change, so this is like one letter in one word, we have a mutation that when we make a mouse model of it, is able to capture the symptoms of SCN8A. So I have mice that have the normal copy and then some that have the mutation, and I record the neurons from both, and then I try to see what are the differences between those, and I assume that the differences I see between them are probably important for epilepsy and are probably due to that mutation. And so, right now, the work that I just talked about, so I've recorded from inhibitory interneurons, which make up 10% or less of your whole brain, but turn out to be really critical for protecting the brain from becoming hyperactive. So to do those experiments, I just record those neurons. I can do different ways of recording to get different type of information. Should I say more about my rig?
I was hoping also that you could talk a little bit about, so that graph that you put together looks an awful lot like things that many of us are used to looking at EEGs,
Let's
right?
see, these kind of?
Yeah, the action potential.
Okay.
Yeah. So that
Yes.
bears some kind of resemblance to things that we're used to looking at. Can you explain? what that
Sure,
is?
I'll do my best. So these are recordings. So I don't have it labeled here, but so time is what's changing as we go in this direction, and then on the y-axis is voltage. So these recordings are from one single neuron firing action potentials. Each of these is a single action potential. Many of you may be more familiar seeing EEG recordings, which is also voltage over time. However, in EEG, you're recording the activity of many neurons. So I'm trying to think if I have another good metaphor. Might be tough, but you can, if you took all of these neurons and they were summed from many different neurons, and you were recording the average of all of these little spikes. In the normal brain, they kind of cancel out because there's so many, they're happening all over the place. But if the brain reaches hypersynchrony and all the action potentials start going at the same time, whenever you record from many at the same time, they begin to have these spike wave format. So that's another way of saying at the population level, many neurons are finding action potentials at the same time. That's another way of saying hypersynchrony, like I said earlier. So during a seizure, the network becomes hypersynchronous. If I were to record individual neurons and stimulate a seizure-like activity, because whenever I do recordings, I can't get a seizure in the slice. What seizure-like activity looks like is that all the neurons just fire big bursts of action potentials all at the same time in a rhythm. So, does that help a little bit, or just try?
Are there any times, just going off your previous statement, are there any times that actually the brain requires hypersynchronization, like learning or remembering, for example?
Yeah. Yeah, you're dead on. And this is such a complicated. The question is, does the brain require hypersynchrony at other times? And the answer is yes. A few that come to mind would be sleep, particularly in I believe it's stage 3. Whatever is the deep sleep where you really need to activate your immune system, that requires hypersynchrony of the brain. So it would be wrong to try to treat epilepsy by completely desynchronizing everything in the brain, because we need that to be coherent. And so that takes me back to that image I showed of the tightrope walker. This is such a precise balance, because as many of you know, in a much more personal way than me, stopping seizures also can stop a lot of other functions. And that's an important piece that, as researchers, we're trying to restore balance. We're not trying to, if our goal is only to block seizures, I think we're not approaching the problem with the correct mindset. Exactly. Another way of saying it is dosing matters, getting the right drug. Like if a drug's mechanism of action just totally shuts down neuronal activity completely, that shouldn't be our main candidate. That's anesthesia. Yeah. So it's a difficult. It's about balance, and balance changes because the brain changes itself. That balance isn't one thing from day to day, and that is an incredibly difficult problem to solve from a pharmacological perspective as a researcher. So ongoing, but it's a challenging problem.
Two more questions.
Could you explain a little bit the gain of function versus loss of function
Yeah.
for folks that might have an interest in that? And then my second question is, could there be variation in phenotype where there's the same mutation? Could it just be due to other mutations, either that have just not been found or a new method like sequencing?
Yeah, I think the second one gets directly at the point we had earlier, is that even though in a research context we're thinking very simply of single mutation leads to single change, that's what we have to do in the lab in order to try to control things. But in the context of an individual with SCN8A, I think it's very complicated with other genes involved. The body's pretty impressive in being able to compensate, but sometimes that compensation can either mask something or it can become a problem. So it's the multitude of genetic factors, all multiplexing is an important question to be addressed and is difficult. The first question, I'm trying to remember, gain of function, loss of function. So that's yes. It's a term that I'm going to add. So this term is used a lot in the literature. One issue that I don't love about the term is that it has a genetic meaning and it has a physiological meaning. And so I'm going to answer your question and say, with respect to physiology, so physiologically, gain of function means that it's simple. It just means the channel is doing what it does more. So it's letting more sodium in. A loss of function would be it's letting less sodium in. But again, I add that caveat that's physiologically gain of function or loss of function. The reason I say that is because I've had discussions with my geneticist colleagues and they use those terms differently. And sometimes I've had at least one occasion where I'm talking about gain of function physiologically and they're talking about loss of function and they're actually similar things. So to keep that proper cycling of sodium channels, you have to have the right balance of sodium that comes in. If you have more, which is, we only determine that through physiological measurement, like what we do in our lab, then we can say it's gain of function. So for example, many of the ones we find, they have their gain of function in that they don't close completely. The channel current stays on rather than completely inactivated.
So how does the action potential look different from loss of function?
Ooh, good question. So the question is, how does the action potential look in a loss of function? We haven't done that experiment. I can't recall if that experiment has been done. Actually, I take that back. That experiment has been done. I'm thinking of the paper now. But it's not in the context of SCN8A. That was a mouse model published that had ataxia. It didn't move well. It's another mouse that Miriam created many years ago called the Med-J mouse, if any of you are interested. But it I believe, had, in the same way that ours had extra action potentials in the slide, I think, previous to this, the loss of function would have fewer. And that's generally the accepted idea of what's going on Dravet syndrome.
Kind of more of a basic question. Can you help me understand the correlation between the SCN8A and the developmental delays? So obviously this has to do with the communication through the neural networks and all that stuff. But how is that actually impacting speech or memory? Because if the kid is not learning anything, that's obviously they can use that at a later point of time. The basic level.
Yeah, I don't know if I can give a great answer other than we know that to have language, to have coordinated movement requires really precise function of neurons functioning in networks. And so if even a small tweak happens in a single neuron that disrupts that proper network function, it will impair speech or learning or movement or sleep. So it's even, I guess the takeaway is a small change in the channel could lead to wide-ranging effects at the level, because there's so many voltage-gated sodium channels and so many neurons that are required to very tightly function. The developmental aspect in terms of like, I don't know if some of you may describe your experience as where it seemed to be developing normally and then regression. Some of that may be due to, in the very early days, very early stages of development, the brain, there's some evidence that the brain is using a different sodium channel, and then it begins to switch on to use 1.6 more. And so that could be part of the story. Although those, I think we need more people looking at the developmental side to answer that completely. I have one way back. Oh, I'm sorry. Sorry. I scared you.
Sorry, so I'm talking on behalf of a parent that is in Texas. So her son, usually, I know the onset is usually when they're babies, right? They're usually, but his started when it was about 12 years old. So his actually development was fine. And he was actually like in high, like academic. So, and then of course the teachers started and then, so,
yeah.
And his was inherent too from mom. So, I mean, I know that at least to us, it's kind of fascinating because, like, I know, like, there are other cases like Jose's dad, Saki's dad, where the carriers, but because, I mean, he would have never known had his Saki had that. You know what I mean? Yeah. So I guess the more research you guys do, like, the more people get on, you can see different variations too.
Exactly.
That, right?
I think every story that gets accumulated gives us more insight. One point I would add, even though the body is constantly, in some ways, the body is constantly developing. So even at age 12, even though it's much later than many, that's still, I think it's fair to say, it's still it could be still developmental. There was a talk last night with folks with Dravet syndrome, and they presented evidence of a patient who was given a different treatment at age, I think it was 60. Were you there last night? And they had spontaneous speech at age 60 upon being given the. So again, it's just a single, these are the case studies. But the point is, I think, we shouldn't underestimate the brain's ability to constantly be adapting and changing. And so studying development is such an important piece, and I think actually development is longer than just early development, but certainly trying to understand why there might be a period of normal development, and then when does it start. This is something we need to understand.
You shared an incredible amount of information today. What should we as parents be looking for tonight in the talks that is so relevant to what you were speaking of?
That's such a good question. That's a challenging question because, so the talks, I mean, I feel like we've covered a lot. I'm really proud of, if you've gotten this much, I think that's a really good start for understanding the talks tonight. I know that talks can be kind of overwhelming. I don't know if any of you felt that. I'll let you know a little hint. Even as a scientist, I feel overwhelmed frequently by the talks. So just do the best you can to try to understand. I would say one place to start is to try to think about what part of epilepsy research are they trying to answer? Are they looking at neurons? And if they're looking at neurons, you'll see recordings like this. If they're looking at patients, then they're trying to understand what's happening at the level of behavior. And so place the level of what kind of questions they're answering, and then think to yourself, maybe a good question is, what are they trying to show me about the balance of epilepsy or neurons. Like, are they showing me that something is changed? Or are they trying to show me a way to bring it back? I think those are the kind of strategies I have when I'm listening to a science talk, is to try to kind of place everything. And if you can if you find yourself overwhelmed and you tune out during a talk, one thing I'd recommend is try to turn it back on for that last slide when they say, in summary, because if you can get that, then you've gotten as much as I think is required to really work with something meaningful. It's going to be challenging. The other thing you can do, although I hesitate to say this in case my email gets bombarded, but you do have specific questions, reach out. If I do get overwhelmed with emails, I'll give it as a task to my undergrads to check their knowledge. But the point is, they're difficult to follow, science talks. This is a good start. Think about the brain as a complex input-output machine that requires balance to do things properly, and then watch for the ways that people will show you that something's imbalanced or how they're trying to bring it back to balance. Is that fair? Do you have any suggestions?
That's great suggestion. One thing about who knows what. I think in some ways, you guys know more than the scientists know, and the scientists end up learning things like things you already knew. Well, yeah, my child gets sick and I get more seizures, or there's some thing that you can correlate with how your child's doing, and someone tries to study that and put it on a scientific basis, and it's like, in the end, you go, well, we already knew that. Then they understand the mechanism, and they might be able to find an intervention or a therapy that is good with that. So, in a way, what I'm trying to understand is collectively what we all know so well about our children, and put that into one new body of evidence, new body of knowledge that can inform the researchers, can inform the clinicians, can inform the industry partners who are trying to get better therapeutics. In the end, you guys know more than the scientists know, and you know everything about your child, that you know how frustrating it is to communicate what you know and have something to be done about it. So that's where things need to go.
I don't have any more. I don't have any more. That's good.
Thanks, Michael. Hi. I just want to thank you, Eric. I know there were just a few families last year who got the benefit of this talk, and that was one of the first items on the list this year at the planning event, was being sure that all of you got to learn from Eric's insight. And we really wanted to let all of you feel like you're going into the talks tonight equipped to follow as much as you can. And also, I think it's really helpful to know that even researchers aren't able to follow all of their colleagues' work. And having learned all of this stuff kind of on my feet as well, and I was still learning from Eric today. So we're going to have some coffee coming in a little bit. We have some time to just chill and mingle before the main event this evening. And I encourage you guys to continue to bombard Eric with questions.
I'm here until, yeah.
10 p.m. We'll be here until 10.
And possibly beyond. Welcome everybody again to this event. You guys hang a little bit tight, and I know there should be coffee coming in. Do you want to say anything, Billy? No? Did you have something to say? No? Okay. So thank you guys again all for being here. It's really touching, and we look forward to getting to know all of you better.